demonstration during laboratory sessions for undergraduate students. And also I've been a graduate research assistant during my studies, where I'll have to perform and also publish my research works. And currently I'm with Bioinformatics Scientific as a product specialist, where basically I'll have to identify leads and market potential, and also provide technical information on the products and service we offer. So these are some of my memories during postgraduate studies. First, during the Malaysian International Seminar on Antarctica, where I was an oral presenter. And then I also presented at X International Conference, which was supposedly held in Ukraine, but too bad pandemic hit us, so we just had it on online platform. Then I did my research attachment program at Japan, and during my research attachment...
But your slide was stuck.
Oh, my slide stuck.
Or you added the front page.
Oh, okay. How about now?
It's still stuck.
It's still
okay. Now okay.
Okay, I think I'll just cancel my presentation. I just use the normal view lah. Okay. So this is my academic and experiences. So then as I said earlier, this was during Malaysian International Seminar on Antarctica, and also at international conference held up and organized by Ukraine. Then I did my research attachment program at Japan, where during my research attachment program, I participated in the symposium on polar sciences in Japan. I was a poster presenter there. Then I had to present my works during my research attachment programs in front of the Japanese lecturers and students. So this was the pictures taken during that time. And currently now I'm with Bioinformatics Scientific as a product specialist. So this is the outline of the presentation for today. I would talk about the next generation sequencing and a little bit on whole genome sequencing. So before going into the topic, I would like to give a basic picture on the sequencing technology. So as you all know, DNA, it contains the blueprints of life. Within its structures are the codes required for the assembly of proteins and non-coding RNA, where these molecular machineries affect all the biological systems that create and maintain life. So by understanding the sequence of DNA, researchers can elucidate the structures and also the function of proteins as well as RNA, which are basically you can get the blueprint of the organisms, what they can do and what are the abilities they have. So what is NGS? The next generation sequencing, or high throughput sequencing, or massively parallel sequencing are related terms that describe the DNA sequencing technology, which has revolutionized the biological research. NGS, it is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. Characterized by the ultra high throughput, scalability, and also speed, NGS enables researchers to sequence the human genome within a week, which Sanger sequencing took over a decade to accomplish the final human genome draft. So let's see the evolution of this sequencing technology. For the first generation sequencing technology, as we all know, Sanger sequencing, it is a method which D-deoxy chain terminating sequencing method that has been recognized as the global standard of DNA sequencing for nearly 40 years. Even after the Human Genome Project, the cost of Sanger sequencing remained too high to enable this kind of large-scale sequencing projects, which lead to the second generation, which also known as the next generation sequencing. But till today, the Sanger sequencing is still in use for low throughput sequencing. For example, you just want to identify the genus level, up to genus level of a species, you can go for Sanger sequencing. So for the second generation sequencing, the most common platform, as we all know, is Illumina. The key innovation of Illumina platform was bridge amplification, which allows the formation of dense cluster of amplified fragments across a silicon chip. Amplification of the original single molecule into a large cluster of many copies is what makes it possible to detect a fluorescent signal as a single DNP is added one at a time as sequencing proceeds by synthesis. Their extreme capacity allows for sequencing at very low cost and more faster. They are limited, however, in length. Like NGS platforms typically produce reads of 50 to 500 base pairs in length in few fragments. So since short reads are not suitable for all sequencing projects, this sequencing method was further developed, which opened the door for third generation sequencing. This technique uses miniaturized wells known as zero-mode waveguides, in which single polymerase incorporates labeled nucleotides and light emission is measured in real time. This single molecule real-time sequencing, like for example PacBio, has ability to produce long reads, like tens of thousands of bases long in a single read. These long reads make it possible to span large structural variants and challenging repetitive regions that confound short read sequences like Illumina, because their short snippets cannot be differentiated from each other during assembly, which can cause overlapping or gaps. Another advantage is this method does not require PCR amplifications, hence PCR bias can be eliminated. So this is all on PacBio, but very recently a new method has been established in the third generation sequencing, which is called the Oxford Nanopore technology, also known as MinION. It is a different approach on a single molecular approach to long read sequencing, which uses pore-forming proteins and electrical detection to do the sequencing. So this is the summary of what I presented before, the evolution of the sequencing. This is how it is done in first generation, second generation, and also third generation sequencing. So each has their strengths and weaknesses. So we have to know our purposes to know which sequencing to do. So this is some of the common platforms used in next generation sequencing. The first one is Illumina, second is Ion Torrent, and also Roche 454 sequencing. These are the sequences we will perform the sequencing. For third generation, these are the platforms. The first one is PacBio, and the second one is the newly launched MinION. So these are some types of next generation sequencing. Like for example, in targeted sequencing, we can perform whole genome sequencing, whole exome sequencing, 16S metagenomics, and so on. For genome sequencing, we can perform shotgun metagenomics, de novo sequencing, plasmid sequencing, and so on. And for transcriptomic sequencing, which mainly focuses on RNA sequencing, like RNA sequencing or small RNA sequencing. So today, my focus will going to be on whole genome sequencing. But I was just showing what other sequences can be done using the next generation sequencing. So what can NGS do? Actually, NGS technology allows researchers to deeply sequence target regions or rapidly sequence whole genomes or specifically sequence transcriptome to identify splice sites and RNA sites. So it also allow epigenetic modification, which is epigenetic modification is actually they alters the physical structure of DNA. For example, like DNA methylation, the addition of methyl group to part of DNA molecule prevents certain genes from expressing. This is how they do the epigenetic modifications. And NGS also allows study microbial diversity and also evolution. And the sequence diseased samples to discover variants, like maybe this can be biomarkers or therapeutic targets. NGS also conveys enormous volumes of information in a short measure of time to help get together of novel genomes. With the reducing price and increasing precision, NGS has become an indispensable tool in genomic research and also translated into routine clinical practice for precise medicine. So some of the application of NGS, next generation sequencing, are applied to different aspects of biological sciences like plant biotechnology, animal biotechnology, and also human biotechnology. In my case, during my PhD, I uses NGS technology in bioremediation, which I will be explaining later. So the plentiful information generated by NGS assists in analyzing genomics variation, various disease mechanism, and also its resistance. NGS has its application in forensic genetics as well, where it contributes to offering new possibilities for forensic genetic caseworks. NGS can be combined with various types of markers like STR, SNPs, which is single nucleotide polymorphism, insertion or deletion mRNA to gather more information from a unique samples in a single experiment. All these sorts of application of next generation sequencing helps in development as well as the advancement of better diagnostic tools in medicine field, pharmaceutical field, bioremediation field, food safety field, and so on. So, on whole genome sequencing, what is whole genome sequencing? And what can whole genome sequencing give us? Okay, this WGS reveals the complete DNA makeup of an organism, enabling us to better understand variations both within and between species. This in turn allows us to differentiate between organisms with a precision. WGS techniques generate from microbial sample multiple short reads that can be assembled based on overlapping regions like de novo assembly or mapped to previously published reference genomes, which then enables the comparison between microbial strains that genetically diverge at a single nucleotide. Such precise identification and classification of microbial strains, as well as the parallel sequencing of different bacterial strains or microbial strains in a single run at low cost with a quicker turnaround time, have made this WGS the most convenient tool for clinical diagnostics and as well as other diagnostics in real time in real time. So this is a simple workflow how it is done from the extraction of DNA to the sequencing. So first, we will culture, okay, in my case, I use bacteria. So we will culture the bacteria. Then the genomic DNA will be extracted from the cultured bacteria, and they will test the quality and the quantity will be tested to see that DNA complies the requirement of the sequencing. So this DNA then is cut into short fragments of known length, either by using enzymes like molecular scissors or mechanical disruption. This sheared DNA then will be copies of this DNA will be made using a process called PCR, as you all know, polymerase chain reaction. The pool of fragments DNA in PCR machine is called the DNA library. DNA library is actually the ligations, fragmentation of the DNA and addition of the adapters to the DNA. Then this DNA library is then loaded into the sequencer. Like I told you before, there are many platforms like Illumina for the sequencing. So the combinations of nucleotide, ATCG, making up each individual fragments of DNA is determined, and each result is called a DNA read. So how we can know what are the sequences? That's where the bioinformaticians come. The sequencer produces millions of DNA reads, and specialized computer programs in bioinformatics analysis are used to put them together in the correct order, like piece of jigsaw puzzles. When completed, the genome sequences containing millions of nucleotides in one or few large pieces is ready for the further analysis. So a little bit on what I did during my PhD program. So my focus was to identify the genes involved in the phenol metabolism, which enables the bacteria to degrade the phenol, which has been polluted the environment. So what I did, I extracted the gDNA from my bacterial samples, and this gDNA I checked for the quality and quantity using nanodrop. They have certain requirements on for the purity and the quantity of the gDNA. Once the gDNA met the requirements, like it has the correct concentration, has the correct purity, we can send for the sequencing. So I sent to the service provider to do the sequencing for me. So I did PacBio sequencing, so it requires a high molecular weight DNA where you can get one contig, means you can get a long read of the sequence. So there they did the library preparation, sequencing, and also they did for me the genome assembly and annotations. So what I did here was data mining. Once they gave me the analyzed data, I can know the species, know the genus and the species of my bacteria. And from the genome assembly and annotations, I can find the genes responsible for the degradation of phenol. So this phenol does not degrade with one enzyme. They involve a pathway of a series of enzymes which call a pathway. So I have to identify the genes that encodes for each enzyme that are responsible for this phenol metabolism pathway. So since my bacteria was already it's not a novel genome, so I had a reference genome. So I have to do the literature review. I have to compare my bacterial species with already published bacterial species, and I have to compare the pathways between them so that I can map the gene and know the pathway that involved in the phenol metabolism. This is what I did during my PhD studies. So these are some of the career options and companies carrying out NGS research. Like for example, if you are really interested in NGS, you can pursue your career as a bioinformatician, technical application scientist, field application scientist, as also research officers with different project titles which involves NGS. So these are some companies, they carry out NGS research mainly. So maybe I guess you guys have not done your internship or final year project, right?
Yes, Doctor.
Hello?
Yes,
Okay.
Doctor.
You are going to do your internship programs?
Yes.
Okay, so maybe if you're interested in NGS, you can find the institute or companies that provide this kind of NGS research so that you can build your skills and you can get the knowledge on this. So later you can use that when you are finding jobs, or you can use that skills during your postgraduate studies. So yeah, this is how it has done. So before completing my presentation, I would just like to give a brief idea on what Biotree offers. We are offering next generation sequencing, oligosynthesis, gene synthesis, and peptide synthesis service, as well as reagents and kits for molecular and protein works. And yeah, so that's all from me. Thank you.
So thank you so much, Dr. Kavilasni, for the informative speech. So, all right, ladies and gentlemen, I think we can proceed with the Q&A sessions. You guys feel free to type out your questions in the chat box, or you can also unmute your microphones to ask questions to Dr. Kavilasni. So all questions are welcome now. So here's one question.
Yeah,
Yes.
in what basis do sequencing
Is price? Is it fixed? And have they changed in the past years?
Yes, they have changed in past years, and the sequencing price depends on which services are you looking for and what are the species. So they differs. If you are performing on eukaryotics and prokaryotics, they differs there. And if you want to go for 16S metagenomics or whole genome sequencing or RNA sequencing, they differs there. And in terms of the price, okay, let me give an example. Okay, when first the third generation was launched, which is PacBio, the price was super high because that was the PacBio was first launched. Then eventually the researchers done, and they done from their part, they done researches to see if they can reduce the cost, or they can they have done the researches. So based on that, the price has been reduced compared to when early they first launched the sequencing technology. This is how it has done for the Illumina when they first launched. during when the third generation doesn't exist, when only the second generation exists, the Illumina price was high. And once the third generation sequencing launched, the Illumina price is slightly reduced compared to the third generation sequencing. So the price will be even they will fluctuate, depends on the research and technologies we have.
So thank you, Doctor. Is there any more questions?
Good morning, Doctor. Does any techniques will replace NGS in the future? Possible. It is possible because no one would have been imagined the next generation sequencing will be replaced with third generation sequencing now. So maybe in future, a third generation sequencing will be, it's not replaced, but they are developed into more advanced technology. So that is what we call next generation sequencing. The next will be continuing next. You know, you understand? Yeah. If there is one gene that experiences mutation, can NGS application be applied for this kind of situation? Yes. As I said before, there are the sequences you can detect the mutations, insertion, deletions from the sequencing using NGS technologies. So like before when I said the epigenetic modification, they actually add DNA methylation to stop the gene from expressing. So that is how it is done. So basically from NGS, you can get the whole picture of a species or a genome. Yeah.
All right, thank you, Doctor. Any more questions from the audience? If not, can I ask a question, Doctor?
Sure.
So what are some challenges or difficulties facing during for this whole genome sequencing?
Do you mean during the sequencing or the whole process, or
The whole process. Is there any challenges, difficulties?
Okay, as a researcher, to get the samples itself is a challenging. Okay, for example, PacBio, they require high molecular weight DNAs, and the concentration should be super high. So if you are using, for example, I will share my experience lab. I used gram-positive bacteria. So to extract DNA from gram-positive bacteria is quite hard compared to gram-negative bacteria. And my bacteria, they have a texture which is mucoid, where you will get the end product is quite viscous. So viscous samples, you can't proceed with sequencing. So you really have to extract the DNA which met the requirements, then only you can proceed with the sequencing. So when it comes to sequencing, there you will face errors like contaminations or during the sequence analysis, the error rate should be, redundancy, repetitions, and so on. So you will have to be very meticulous when you perform the molecular analysis. That is where the challenge lies. Yeah.
Okay, thank you, Doctor. Any more questions, guys, from the audience? You can ask from Doctor now. Okay, there's a question from Hui Yi. Good morning, Doctor. May I know the limitation of NGS?
Okay, limitation as in, you mean in the whole picture or okay, I'll just explain both. Limitation of NGS, now to perform the NGS, you will need the technical experts and the technology, and you need the platforms. So for that does cost high. If the NGS platform itself is cost high, and then to perform the analysis and the bioinformatic analysis, you require a technical expert there. So that is where the limitation of NGS lies. And in comparison with the next generation sequencing and third generation sequencing, next generation sequencing, they have the PCR bias, where we'll need to amplify the DNA prior to the sequencing. This causes the what they call PCR bias. So that is the limitation of Illumina sequencing. That is why the third generation sequencing was done. But then third generation sequencing, they have the limitations of error rate, where the error rate of the third generation sequencing is quite high compared to the second generation sequencing. So continuous research is going on to improve these technologies actually. May I know how long the genome sequencing can be done for a sample? Okay, for Illumina sequencing, maybe in a week. For PacBio sequencing, you can get it in 10 hours or 12 hours. The sequencing, just the sequencing. Then after that, the analysis, that depends on the bioinformaticians. How do we choose between NGS and third generation sequencing? Okay, for this, if you really want long read sequencing with no gaps, where you can get a complete data on the sequencing and you have the budget, then you can go for third generation sequencing. But if you're just working on bacterial samples or low the data which is not as high as human or eukaryotes. Okay, prokaryotes have less data compared to the human, right? So when you use bacterial samples or so, and you don't need in detail data, then you can go for NGS, and it is much affordable. If there is a pregnant woman that has been detected to have mutated genes, such as Down syndrome, can NGS used? Yes. This this technology is currently they are developing in China, because China has spent a lot on molecular diagnostics. So they are actually working on this to use these NGS technologies in medical field. But currently it is in a clinical trial phase. So once it is launched, then maybe we can even detect the disease in every single species in Earth. Actually, they are doing a project which called Earth sequencing project, where they are planning to sequence each eukaryotes on Earth. Actually, yeah.
All right, thank you so much, Dr. Kavilasni, for the Q&A sessions. That was so interesting questions just now by the floors. So I think that's all from Dr. Kavilasni session, and we can move on to the next session. So Shafika, you can take the floor now. Shafika?
Alright, thank you, Aiman. So, hi everyone, my name is Shafika. So, we will begin our second session for today. By the way, that's such a awesome Q&A session just now. So, now let's continue with our second outstanding speaker, technologist Dr. Nur Azlan bin Nur Muhammad. So, Technologist Dr. Nur Azlan holds a certificate in Bachelor of Biomedical Engineering, majoring in Bioinformatics in 2008, and a PhD degree in Bioinformatics from the University of Melbourne, Australia. Currently, he is the head of Centre Food Bioinformatics Research (CBR) at the Institute of System Biology, or also known as INBIOSIS, in University Kebangsaan Malaysia. His expertise in bioinformatics and computational system biology. His research focus is on the application of omics technologies to unravel the new genes and pathways for the development of precision biotechnology. He applies genomics, transcriptomics, metagenomics, and metatranscriptomic technologies based on the short and long reads of next-generation sequencing platform. Hence, this workflow are then combined with the sequence analysis, domain characterization, phylogenetic pathway reconstruction, and also database development to unravel the new knowledge from the big biological data. So now, I welcome Dr. Nur Azlan to share his knowledge on the topic of next generation sequencing accelerates system biology research and application. So, Dr. Nur Azlan, here's the floor. Thank you.
Okay, thank you, Shafika. Morning, everyone. Morning, Dr. Kavilasni. Kavilasni, sorry. Is my voice loud enough? All right. Okay, very clear. Because I don't have a headset today.
Yeah, clear, Doctor.
Right, okay. Let me try and share my slides. Right, morning everyone. Assalamualaikum. So thank you for inviting me for this session today. So I believe you all are microbiology students, right? So I believe everyone's very familiar with what are genes, what are proteins, and so on. So today I'm mainly going to focus on explaining what is systems biology and how we apply NGS as a tool to do systems biology research and the way forward roughly in systems biology. So here are roughly my contents for today's talk. So first, let me introduce where I work. So I'm from the Institute of Systems Biology, or INBIOSIS. I'm located in Universiti Kebangsaan Malaysia, Bangi campus. So we are quite near, only 15 minutes from UPM. If you guys are interested, feel free to contact me if you want to come and visit us. So basically, INBIOSIS was established in 2005. So 17 years already. So we have 3 centers. At our institute. The first one is Center for Genome Analysis and Technology, or CGAT. Second one is Center for Plant Biotechnology, and lastly, Center for Bioinformatics Research, which is the center that I'm heading in INBIOSIS. So this is the view of our facilities. On the top left is basically the main building. So it's where the administration is located. And 4 of our labs are located here: our genomics lab, metabolomics lab, proteomics lab, as well as bioinformatics lab. On the right, we can see complex rumah tumbuhan, and also at the bottom left. So this is our basically Center for Plant Biotechnology labs. So we do tissue culture there. We plant some of our plants for research there as well. So at INBIOSIS, we have a few research focus. First is to use biotechnology to basically analyze our local biodiversity. So here are some of the plants that we are working on. We have Genome, our flagship project, which basically initiated in our institute. We also have a few research working on kratom, mangosteen, as well as pitcher plant. We also want to contribute in how we can help the food security of Malaysia. So we also work on rice, some of aquaculture, such as grouper as well as prawns. And lastly, we want to venture into developing sustainable bioproducts. First is for example, cocoa disease control, oil palm disease control, as well as some essential oil products that we gather from our plants that we are working on. So basically, my work mainly focuses on the disease control of oil palm. So I work on some of the species that are currently affecting the oil palm trees. Either both fungus disease causing as well as some insects that hits the oil palm leaves and damages the tree. In terms of our research scope, so what kind of projects or what kind of analysis and research we do onto our species of interest. basically we apply omics technologies. So here are some of the machines that we have in our labs. So in terms of facilities, we are very capable in doing metabolomics. So for the other platforms, proteomics as well as NGS, specifically, we don't actually have those facilities here, so we outsource them basically to company like Biotree, for example. Then from the data we obtain from these omics technologies, we basically apply computational systems biology, and from the findings, we do some in vitro work, we do some synthetic biology, and hopefully we can basically apply this knowledge gain towards solving problems that are faced by the industries. So what is systems biology? So there are a few definitions. So here I quote here 4 definitions. So basically systems biology studies biological systems. So it actually came from an engineering concept where people look at systems as a whole. So in systems biology, we basically see the biological system as a whole, as a big picture view. So we look at the set of genes, proteins, as well as the pathways related to this biological, we call them as entities or features as a whole. So instead of looking at one single gene, how a single gene works, how a single protein works, we look at a group of them and try to figure out together how they work and how they affect the species itself, the functions, the development, or so on. So here's a visual representation. If we look for that, we display this biological system. So on the left, it actually Tokyo subway route map. So basically, we can see how the different stations of the Tokyo subway are related to each other. So for example, we want to go from point A to point B. So we look at the map and we know where to go, where the station is located at, and so on. So basically, systems biology uses omics dataset, and basically we map out the information gained from the omics technologies and try to figure out the relationship between genes and proteins and put it on a map. Why this is important? Basically is to give a better understanding. For example, just now people mentioned about Down syndrome and so on, right? So in Down syndrome, for example, we know what chromosome are affected. We know roughly what genes are affected by the mutation. But how does the mutation affects the workings of the human body? So that's why the map, the interrelations between the genes is important for us to know, so that we can know and predict what are the effects of certain mutations. So from the understanding, we can either treat, or we can either prevent, or we can either make diagnostics better as a whole. So at INBIOSIS, we also do synthetic biology. Basically, from the systems biology, once we figure out what are the pathway or the genes are related to certain pathway, in certain cases, we would like to test whether that pathway, what happens if we, for example, change some components of that pathway. So we use the synthetic biology platform where we transfer some of these genes into lower complex organisms such as bacteria or fungus, and try to basically replicate the pathway that we are interested in understanding in those simpler organisms. So that's one. We also have some work where we try to produce some compounds that are being produced by this pathway basically is to mass produce those compounds. So instead of growing the plant, which we need to water it, we need to give fertilizers, we need to spend a lot of time on it. So instead of getting the compounds from the plants itself, we would like to just grow bacteria, and that bacteria is able to produce the same compounds. So first, we need to basically create a new bacteria that can produce those compounds using the synthetic biology approach. Here, I just want to highlight the central dogma of biology. So I guess everyone should be aware of this. We have DNA, we have mRNA, as well we have proteins. So for NGS, it applies to the first two, which is the DNA as well as the mRNA. So the nucleotides basically can be read and converted into digital data so that we can do bioinformatics as well as computational analysis to figure out what are the genes being expressed and so on, what are the mutations, what are the SNPs and so on. Here I want to highlight what are omes. So from the introduction of NGS as well as other omics technologies related to the NGS, such as metabolomics and proteomics. So basically now we can profile the entire what we call as ome set, the genome, the transcriptome, the proteome, as well as the metabolome. So what's the genome? Genome is basically the set of all genes from our sample. Transcriptome is the set of all RNA, so it's also called RNA sequencing, RNA-seq. Proteome are the set of all proteins, and metabolome is basically the set of all metabolites in the sample. So everything that ends with OME is basically the data set. Everything that ends with omics is basically the technology, the platform. So we have genomics, we have transcriptomics, proteomics as well as metabolomics. And in terms of the technology, basically NGS covers the first two ome, the genome and the transcriptome work, because both of them are basically ribonucleic acids, which can be read and converted into basically sequencing just produces text file. So all your nucleotides are being represented by the letter A, T, C, G, C. Okay, next I want to talk briefly about next generation sequencing, since Dr. Kavilasni have mentioned a lot about this, so I just have a quick through. So actually this technology is not that new. It's over about 50, 60 years old already. But NGS basically came in the early 2000s. So 2002 we have the complete mouse genome. Then we have all the NGS technologies, the Illumina. Then from 2010 onwards, we have the third generation, which are the long read sequencing, which is PacBio. Now we have PacBio Hi-Fi. And one of the latest one is Oxford Nanopore, which basically can read the entire nucleotide strand without chopping it. So basically, to my understanding, there's no actually limit. So the limit is just your sample. So in next generation sequencing, millions of reactions are performed in parallel. So we have the term as short read sequencing as well, because the reads are quite short, 150 to 300 base pair in length, compared to the millions of nucleotides in the whole genome. So basically, this large genome is being chopped into small pieces like a puzzle. So from that, each component of the puzzle is being read by the sequencing machine. Illumina, for example, if you choose Illumina as the platform, then you get your short puzzles, and you need to do bioinformatics to basically assemble back reads into the complete or draft genome of that species. So in terms of development, we are now at the third generation sequencing era, where people are basically trying to develop sequencing tools that can read longer and longer reads, so that we don't actually need to do a lot of the completing later. So we don't need to do a lot of assembly. We don't need to figure out how to assemble the big puzzle that we have in the NGS era. So basically, as like a puzzle. So if you have smaller pieces, so you have a lot of your number of pieces. You have a lot. So it's easier to assemble a puzzle with less puzzle pieces compared to the one with let's say 1,000, 2,000 pieces, right? So that's why people are investing into longer read sequencing technologies, so that we can reduce the number of pieces, so that the processing later can be much simplified and we get more accurate data. So this is just a list of the different companies and the different technologies. So how they do the sequencing, either by fluorescence or electrical current and so on. And those are basically the references where you can look into the sequencing machines that are being provided by each of the companies. So why there are so many companies? So basically they are competitors to each other, but to me, each platform has its strengths and weaknesses. For example, Illumina is very cheap. Nowadays, if you send for Illumina sequencing, one sample is less than 1,000 ringgit. So it's a cheap way of getting NGS data. But as I said, the reads are very short, so it doesn't actually work well with, for example, my species of interest, which are insects, which has a lot of repetitive sequences in their genome. So that's why long read sequencing technologies also exist. For example, PacBio and Oxford Nanopore. So they basically cater a different kind of problems, but they are much more expensive. So PacBio, I spent 20,000 ringgit on it just to get a single genome data for a single genome. So here are the differences. Basically, the main differences are in the read length. So the shorter read length tend to be much cheaper in the cost. But as I said just now, the bioinformatics part becomes complicated, and so it's a balance, and it depends on what you want to find in the data set. Here are some of the comparison between the different throughput of these sequencing machines. So on the going up is basically how many gigabases per run they can generate, and going to the right is basically how long is each read. So the best is actually the top right there. So it means that we can get more reads per run as well as longer reads. So basically they are pushing that way forward. So you can see roughly here all the different so each color represents products from a specific company. For example, the one in green is from PacBio. So the one in purple is Illumina. So you can see that they are pushing towards that region. So the throughput is going up, as well as the read length is getting longer as well. So this is important for us to do easier systems biology. So how big are these instruments? So basically, this is a snapshot from the Norwegian Sequencing Center. So they have all platforms. They have Sanger, they have HiSeq, Illumina, they have INBIOSIS, they have PetBio as well. These machines are some are quite small, so some are benchtop, some are quite large. For example, the PetBio is quite big. Here are the machines from Illumina. So they have different classes of machines. And so basically, if we talk to the application scientists from Illumina, they will basically give you advice which system to use, which system to have your sequencing being done on, based on what application they're going to do. So on the left there are many different things that can be done in using NGS. So you can do whole genome sequencing, you can do exome sequencing, targeted gene sequencing, and so on. Here are the different machines from the third generation sequencing. So Ion Torrent is not popular anymore, maybe due to the cost and so on. But PetBio is quite being sought after. So they generate a lot of data. The latest genome that I did was about 500 gigabyte in the raw reads are about 500 gigabyte inside just for one sample. So just to highlight how big are these data sets. We also have Oxford Nanopore. So this is a very small sequencing machine. So it's just as big as your external hard drive, and can be basically transported to anywhere. So this is being used in environmental metagenomics, for example, where people go to ponds, people go to plantations and do the sequencing on site, because you just need the device, which is very portable. You can fit it inside your pocket, as well as a laptop to do the sequencing, to read the sequencing data. Okay, next I want to go to how do we use NGS as tools for systems biology research and applications. So this has been mentioned by just now in our first presentation. So genomics is basically the study of the whole genome. At INBIOSIS, we have, I think, already 2 genomics projects. Where we our aim is basically to get the draft genome as complete as possible, just to figure out what are the genes so that we can do gene targeted specific applications for future product development. Then we have transcriptomics, where we look at the differential expressed genes to figure out since we have so many genes in our species, in ourselves as well, but not all of the genes are being expressed at one time. So using transcriptomics, we can basically figure out what are the group of genes that are being responsible for a certain physiological condition. For example, if rice is under drought, what genes are being upregulated or is downregulated, so that we can figure out how to develop or modify the plant itself, or how to give it treatments and so on, in order for it to overcome the problem being faced by the plant and or species. NGS can also be used for metagenomics work. So compared to genomics, where the sequencing is being done on a single species, so metagenomics is basically you try to get the whole genome data from a community of species. So mostly these are either bacteria or small soil microflora. So we also have the term microbiome and so on. So if it's inside the gut, for example, there's basically is a work where we try to get the list of species of bacterial communities. Is either in soil, in gut, in oral. We have oral samples as well. People who do work in the dental research and so on. And from this profiling of bacterial communities, we can basically figure out what are the bacteria that contribute to certain disease or become biomarkers in the environment and so on. So this is one of our projects that we apply NGS on. So Kesum. So this is the herbal leaf that we eat with our laksa. So we applied already 3 omics on this, metabolomics, transcriptomics, as well as proteomics. And this is, for example, the output of the transcriptomics part. So we are talking about millions of reads. So the leaf raw reads are 192 million reads. So from that, basically we need to assemble them. And from the assembled transcriptome library with the annotation to figure out what are the genes and pathway that are present in Kesum. So to visualize what we found, this is one of the pathway, which is a flavonoid biosynthesis pathway that we are interested in, because one of the features of Kesum is very high secondary metabolites, and we also found that this is good. The compounds being produced by Kesum has some medicinal properties such as anti-Alzheimer. We also found that it deters away insects, so it's a good insect repellent as well. And we are interested in this pathway because it ties back to the synthetic biology, because to grow Kesum in a large amount, it takes time. It takes a few months. So you need to have a proper greenhouse area and so on to prevent the attack of insects, diseases. So it's very complicated to grow a large amount of Kesum. So if we can engineer a bacteria that contains this flavonoid biosynthesis pathway, so it's much easier to grow bacteria, and we just extract the metabolites, the flavonoids from that bacteria culture. So it's a similar strategy being used for insulin production. So instead of extracting insulin from animals, we hatch it, which is very costly, as well as the issue of halal and so on. So nowadays, insulin are being basically being produced by bacteria. So we just culture them and extract from the bacteria culture to get the compounds of that you want. So that's for Kesum. Next, we also do some work on Metisa plana. So Metisa plana is we also call it as bagworm. So it's an insect which has a bag around it for it to protect itself. It's very small, but it during its larva stage, it eats the leaves of oil palm trees. And so you can see on the right there, there are holes in the leaves. So this basically slowing the capacity of the tree itself to produce a lot of oil palm oil. So what we did is basically do transcriptomics on each of the stages of the bedworm. So we have data from the egg, from the larva, from the pupa as well as the adults, and try to figure out using transcriptomics approach what are the genes that are responsible for it to develop from the egg into the adult. Why? Because we intend to target some of these genes basically to create control methods or to create biopesticide in order to kill off this bedworm because it's very damaging to the oil palm tree. So here is the bioinformatics that has been done onto the transcriptomics data. So it's a bit different from our Genome work, mainly because the tools have been updated, the databases are much more newer nowadays. So this is about 10 years after the Genome project. And this is totally Illumina. So compared to the hybrid sequencing that we did for the Genome project, this is entirely Illumina. So here again, the data set is quite big. So in terms of the reads, over 200 million of clean reads. So we did our bioinformatics in-house at INBIOSIS, mainly because to give a better understanding towards our own data set. Here are some of the results that we found. So basically there were thousands of differentially expressed genes. So in order to figure out how to get an easier understanding of it, we did clustering. So this clustering is basically groups, sets of genes based on their expression profile. So the idea is the genes that are being up or down regulated as a group should be really interrelated to each other. So we now focus on each of these clusters in order to get better understanding and whether certain genes can be targeted to be developed into control methods. Last year, just want to show how actually everyone can do your own NGS projects. So every time we do an NGS project, so when we want to push for publication, for journal publications or any other publications, we have to basically upload our raw NGS reads onto a few of the database. So this is one of the database that houses this raw sequencing reads. So this is the NCBI. It's an institution located in the United States. And this is the SRA, which is the Sequence Rate Archive database. For example, if you are interested in oil palm, so oil palm alone, we have over 3,000 raw reads. So these are raw reads. So you need to select the ones that you are interested based on your hypothesis and basically do your assembly and do your downstream analysis using bioinformatics. And bioinformatics is very diverse, so many things can be done, and these are example softwares that are totally free to use. So Trinity is for doing the transcriptomics work. We have Cytoscape to do all the network systems biology and so on. So these are basically tools. So the data is public, free. You can download any kind of data that you want from the SRE database, and the tools are free as well. They are open source, so you can get them as well. And next, we also have free computing resources to do this bioinformatics. So here I just want to highlight the Galaxy project, and they have 3 publicly available servers that you can do this bioinformatics analysis freely. So you just open an account, you get quite a strong machine there, 8 core and 250 gigabyte of storage. to do your assembly, to do your differential spreadsheets, analysis, and so on. If you want to get to start on learning how to do NGS projects on yourself. So I will end with our INBIOSIS corporate video. So can you guys hear the sound?
Suatu istilah
Yes?
Yes, Doktor.
Okay.
yang sentiasa dikaitkan dengan kesiasatan forensik dan ia merupakan antara elemen utama dalam penyelidikan biologi moden. Tetapi pernahkah anda terfikir DNA ini adalah merupakan sebahagian daripada data biologi yang memainkan peranan penting dalam perawatan penyakit tumbuh-tumbuhan dan boleh membantu mengatasi masalah ekonomi negara? Sehubungan itu, sebuah institut penyelidikan telah diwujudkan di UKM untuk mengkaji dan memanfaatkan bidang yang menghasilkan lambakan data biologi. Ditubuhkan pada tahun 2005, Institut Biologi Sistem yang lebih dikenali dengan INBIOSIS mengorak langkah mendepani bidang bioteknologi moden di negara ini dengan wawasan utama untuk memantap dan memacu penyelidikan biologi sistem dan biologi sintetik. INBIOSIS memacu kajian terkehadapan melalui 3 pusat percemerlangan dengan menggunakan teknik terkini dalam bidang biologi, sains komputer, teknologi maklumat dan sains data. INBIOSIS membawa era baru dalam kajian produk semula jadi negara dengan mengaplikasi teknologi omik dan biologi pengkomputeran untuk memantapkan bidang phytomixeutical dan data raya biologi dalam mengurai pelbagai misteri sifat biomolekul yang berpotensi dalam pelbagai industri. Juga mempelopori bidang biologi sintetik dan penyuntingan genom di negara ini ke arah penyelidikan translasi dan pembangunan bioproduk. Untuk memacu inovasi ke tahap yang lebih tinggi, INBIOSIS turut bekerjasama dengan industri dalam dan luar negara.
We have been working closely with INBIOSIS since 2015, where we conducted fungal metabolomics.
We have developed a species metabolite relational database called ABSAP, and also we have developed several novel algorithms for extensive analysis of key biological data.
Where we exchange expertise in natural products, phytopharmaceuticals, and metabolomic studies.
In this collaboration, we have identified fungi that are able to live solely based on lipids.
As one of the first systems biology institutes, I believe that INBIOSIS provides a well-structured and strong platform in omics research, especially for young scientists.
Berbekalkan budaya kecemerlangan dan prospek kejaya, INBIOSIS menawarkan program siswazah bertaraf antarabangsa untuk menghasilkan saintis biologi alaf baru.
Di sini, kami diberi pendedahan dan latihan dalam biologi sistem dan biologi sintetik.
Saya memilih INBIOSIS kerana institut ini mempelopori kajian inovatif yang berteknologi tinggi dalam bidang omics di Asia.
INBIOSIS adalah satu-satunya institusi di Malaysia yang mempunyai kapasiti pemprosesan data biologi yang tinggi.
Saya telah didedahkan berbagai teknik makmal yang membolehkan saya menjalankan kajian untuk menjawab soalan-soalan penyelidikan.
Saya dapati INBIOSIS telah menyediakan kesekitaran pembelajaran dan pendidikan yang kondusif.
INBIOSIS memberikan peluang untuk kami menjalankan pendidikan rentas bidang, terutama di dalam bidang bioteknologi dan farmaceutical bagi penghasilan bioproduk.
Sertailah INBIOSIS untuk sama-sama mengilham harapan, mencipta masa depan dan mencapai visi dan misi UKM.
with that, I thank you for your attention.
Alright, thank you, Doctor. Such an informative speech. So, alright, ladies and gentlemen, now we will start our Q&A session. So, all questions are welcome. And as I can see, there's a question at the chat box right now. Okay. So, from Amar Faiz. Morning, Doctor. May I know what is the next project of INBIOSIS?
So basically, INBIOSIS, we have different projects going on in parallel because each of our research fellow focuses on the different kind of scope. So as I showed just now in the scope, we have people who work on different species. So each of the species has their own basically principal investigator and apply. They tend to focus on their species of interest and they apply the various omics towards their project towards their species. So in terms of the next projects, so from my side, I'm focusing on the insect pests that are affecting the oil palm trees. So from the Metisa plana project, for example, so we already done the transcriptomics. So now we just obtain our data for the genomics, and we also have another student who working on the proteomics. So the aim is basically to get this 3 or 4 omics data set, and we want to do multi-omics integration in order to identify the best candidate for pesticide development. So that's some of the highlights lah, and way forward on the things that I'm doing personally. So there's one question there. Besides portable environmental site visit, what the advantage of Oxford Nanopore? Basically, Oxford Nanopore, the focus of the technology is for on-location sequencing. So they develop their product to become a sequencing platform that basically can be done on site. So why this is an area that is very important, because so in conventional NGS or third generation, for example, PetBio, basically you need to get your sample, you need to have a certain purity, then you do the extraction, and you basically have to freeze it because the sample gets degraded in transport. So depending on where the sequencing center is, for example, my sample was sent to the United States. So it takes around, I think, a few days just to arrive there, then another few days on the in the lorry or whatever, and so it takes quite a while. So there is a chance of quite high chance of degradation just going to the sequencing center, and that's exclude all the handling errors and so on, right? So for this kind of work, you need very high, good quality genomic or RNA to get reliable data. So, but for the on-site, basically you significantly reduce the possibility of this degradation lah, because you straightaway take the sample and you put on the sequencer. But the cons is, of course, since it's a small machine, so the throughput is very low. You can only do one sample at a time, and the cost gets high as well in terms of per sample, because you only do one thing at a time, right? So that's the main differences lah. Next share the progress and development of docking simulations carried out in the lab. So we at INBIOSIS, we use docking as well as molecular dynamic simulation, basically to give a better understanding of the proteins that we are working on. So we don't do any of the development in terms of the pipeline, the analysis, or the tool itself. So we use GROMAX for this, for example, to do the molecular dynamics, and we use Autodoc as well as HEADOC to do all the docking work. So in terms of the tool, we depend on them, meaning that the developers. But in terms of our project here, basically we have, depending on the principal investigator, there are certain proteins or whatever they want to figure out the structure. For example, my collaborator, Dr. Maizum from INBIOSIS as well, she wants to figure out what are the possible inhibitors for these proteins of interest, just to filter out the potential inhibitors so that she can do the wet lab validation later, just to reduce the number of testing that needs to be done in assay and so on. With the internship program, yes, just email to me or to the director if you are interested in doing internships here. So yeah, we are open throughout the year. Any more questions?
Doctor, I think there's one more question there, which is there any limitations in doing your project here in Malaysia?
The main limitation is, of course, funding. So as I mentioned just now, omics technologies are quite expensive. So for the PAC bio is about 20 to 30,000 ringgit just for one sample. And depending on your quality of sample and so on, you might need a few rounds of sequencing. So the cost is quite prohibitive because of all these technologies basically came from overseas. So at the moment, our ringgit is quite weak as well. So due to the exchange rate, the cost gets quite a bit higher. Then the next limitation, I would say, because I'm doing bioinformatics, so basically just the amount of computing resources that's needed to analyze these millions of data sets is quite large. So the transcriptomics project, for example, it cannot be analyzed on laptops. It can't even be analyzed on certain desktops. So you need high-end workstations at least to do your analysis. Of course, there's an option of using cloud and other service providers, but that includes cost as well, right? So it ties back to the problem of funding. The next limitation, I would say, just the number of expertise. So as a bioinformatician here at INBIOSIS, we currently have three people who are doing bioinformatics. But places like Singapore or the NCBI and so on, they have hundreds of bioinformaticians just at one place. So we have 3 compared to 200 or 300 people at other research center. So the number of expertise is quite limited as well. So yeah, that's the main challenges. But there are a lot of opportunities as well. Since there are not many people, so you can develop yourself. So you can penetrate the field if you have the right skills and so on.
All right.
Does Autodoc and HEADOC differ? So basically, it's a different tool. So in terms of software, it's a different software. So since it's different, the underlying algorithm is different as well. So depending, I'm not sure what's the exact algorithm, but basically it's like Photoshop versus GIMP. So it does the same thing, but the way it does it is differently. So in bioinformatics, there are a lot of analysis that are being provided by many tools, meaning that there are different versions of the tools. The reason is everyone is trying to create a better software, a much better software, because for example, in docking, biological systems are very complex to figure out the interaction. So everyone is trying to have a better results from their own software and tools. Try to mimic the biological system as closely as possible. So it's a race So that's why there's different tools. Yes, I hope that answers Advantages depending on your data So you need to do your own testing. So some data set are better for Autodoc, some data set are better for HEDOC, because again the algorithm is different, so the results are a bit different. So depending on how your data set looks like, so it might be better for a certain algorithm So if people who focuses on bioinformatics, we tend to compare So if we have 4 tools or whatever, 5 tools that does the same thing, we use all of them and compare which have the best results.
Okay, all right, doctor. I have a few questions. First is there any limitation in doing your research in Malaysia? And second, is there any possibility of a single bioproduct to be applicable to all fungi disease or fungal disease?
Okay, limitation I think I just mentioned just now. So mostly funding and expertise. That's the main limitation But there are too many things to do, so people can basically be involved in doing this kind of work. Secondly, about a single bioproduct. So it's possible, like the way we do drug development. So for example, any medicine that we have on the shelf, right? So it's not specific to Malays or Chinese or Caucasians, right? It's applicable to all humans. So it's the same way in fungal disease or whatever. If we can find a certain target, either protein targets or gene targets that are present in all the fungus that we intend to target, and then it's possible to develop a product that specifically affects all the fungus that you are interested in. But it goes back to how you want to find that out. So basically the current methods is basically to do the genome sequencing of all the fungus of interest, right? And like the way we do human sequencing. I think now the latest project, and they are trying to do genome sequencing of 500,000 people. What is it? No, sorry, 2 million people. 2 million people to basically to find new drugs and because now we tend to see a slow progress in terms of the outcome of new drugs, right? So they are doing genome sequencing on that to solve that problem. Okay, I think you're muted.
Assumption. Hello? Can you hear my voice?
Yes, now I can hear.
Oh. Okay. I repeat my question back. So, since there are many fungal disease in plants, so what are some challenges to produce a bioproduct in order to control this disease?
So the challenge of developing a bioproduct is to figure out what are the target, what genes or what proteins that you are targeting. So that's the challenge number one. So I think even fungal, they have 20,000 proteins or something like that. So trying to figure out which out of the 20,000 odd candidates are the best candidate for you. That's the first challenge. The next challenge is basically to figure out what compounds that you want to be present in your bioproduct. So in terms of the database, I think there's a few million compounds lah. That's just in existing databases. So it's not including compounds that you can produce synthetically. So a few thousand candidates, and another data set is a few million compounds lah. Just to figure out which match to each other is a huge challenge lah. So it's not possible to do it exhaustively. So before omics technology exists, people basically test lah. So take your fungal that you want to target, then take compound A, then you test, die or not. Take compound B, compound 3, blah blah, and right until the end. So nowadays costs are quite high. So it's much, although omics technologies are expensive, it's still cheaper than doing all these bioassays lah manually, one by one. Yeah, that's the way I see it lah. That's the challenges. Yeah. Okay. Hope that answers the Shafika's question.
Yeah. All right, Doctor. Thank you for such an informative answer from Doctor. So, ladies and gentlemen, finally, we have come to the end of this forum. So we would like to thank for both of the speakers for such an informative and interesting talk. So hopefully the forum will be beneficial for everyone. So we will also like to wish for successful findings and output for the ongoing project, Dr. Azlan and also Dr. Kavilasni. Before we end our webinar, so let's take a photo for our memory, and yeah, I hope everyone can switch on your camera for this photo session. So everyone, I hope that you can turn on or switch on your camera. Yeah?
So from what I can see here, we have recorded about 56 participants. So thank you so much everyone for joining this webinar. So Kalista, are you ready?
All right, everyone, ready.
I'll take it in 3, 2, 1, smile. Okay. Okay, one more. 3, 2, 1, smile. Okay, done.
Okay, thank you, Kalista.
Okay, so attention to all participants. Please fill in the Google form that will be sent in the chat box later for attending this webinar and for redeem your e-certificate later. So thank you so much everyone for attending this webinar today, and thank you to Dr. Azlan and also Dr. Kavilasni. Thank you.
Thank you.
With that, so we will end our webinar session. Thank you so much. Also, I wish for both of the speakers and also all the audience that attend our webinar. So I hope that we can meet soon in the future or any future events or webinar. So I am Nur Shafika Falwangi. So my partner.
And I'm Aiman Adam. So thank you everyone. Have a nice day.
Bye. Thank you, everyone.
Thank you.
Thank you, everyone.
Thank you all. Bye.